Process of meiosis.
Let's begin with the preparatory phase, that is the interphase first. This cell goes through three phases, namely G1 phase, S phase and G2 phase in order to grow and prepare itself for the actual division.
And can you tell me what happens to the genetic material during the interphase? That's correct. The DNA is replicated to make an identical copy of each chromosome. On similar lines, other organelles of the cell also get duplicated in this complete phase. Among all the organelles, the duplication of one organelle is equally important as the replication of DNA. And which organelle are we talking about? Yes, it's the centrosome.
And why is the duplication of centrosome so important? Because this organelle contains centrioles which form the spindle fibers needed for separating sister chromatids later. Now, this perfectly grown cell is all set to divide and enter the meiotic phase.
According to the order, the cell will undergo karyokinesis and that will be followed by cytokinesis. Let's dive deeper into this world of meiosis. Meiosis is carried out in two rounds called meiosis 1 and meiosis 2. Why is that so? That's correct. To obtain 4 haploid cells with half the number of chromosomes compared to the parent cell. Let's begin with meiosis 1 first. Meiosis 1 is divided into 4 stages. They are prophase 1, metaphase 1, anaphase 1 and telophase 1. We will list down the changes observed in the cell during these stages step by step. Let's begin with prophase 1. It's the longest phase of meiosis 1. The phase is further divided into 5 substages. They are leptotene, zygotene, pacotene, diplotene and diakinesis. Leptotene is the first substage. In this phase, the cell begins condensation of the chromatin, making it short and compact. The next stage after leptotene is the zygotene substage. Here, the homologous chromosomes form a pair called the synapsis. The new structure that is formed after the union of 2 homologous chromosomes is called a tetrad or a bivalent. After forming bivalents, the cell enters the next substage called the pacotene. Here, the bivalents exchange gene segments between the non-sister chromatids of homologous chromosomes. And can you tell me what this process is called? That's the right answer. It's called crossing over. And the location where the exchange takes place is called chiasma. After successfully interchanging the genes with one another, the cell enters the next substage called the diplotene. In diplotene, the bivalents unbutton themselves, resulting in the separation of the 2 homologous chromosomes. But here, the tricky part is that the homologous chromosomes are still anchored at the chiasma. Finally, in the last substage called the diakinesis, the homologs get completely unhooked and regain their individual identity. The nuclear membrane and the nucleolus completely disappear by the end of prophase 1. Now let's move ahead with the next stage of meiosis 1, that is metaphase 1. The homolog pairs align on the equator of the cell with random orientation. This arrangement is called the metaphase plate and is a result of independent assortment. In the successive phase, that is the anaphase 1, the homolog pairs separate from each other and move towards the opposite poles. Finally, the cell undergoes the last phase, that is the telophase 1, along with the first cytokinesis. The 2 new cells obtained are now haploid in nature. The telophase 1, followed by cytokinesis, helps in the formation of a nuclear membrane surrounding the chromosomes and a nucleolus as well. The daughter cells are haploid, which are formed from the diploid parent cells. Chromosomes, however, are still seen attached to their sister chromatids. So, in order to separate the sister chromatids from each other, each haploid cell undergoes another round of division. But before moving ahead, the haploid cells enter a small resting phase called the interkinesis. It's a short phase with no activities involved. After going through this phase, the cell is all set to undergo the second round of meiotic division. The next round is called the meiosis 2. It also includes 4 stages. In other words, meiosis II can be considered as the mitosis of haploid cells. The first phase is the prophase II. Here, the nuclear membrane and the nucleolus begin disappearing. The chromatid condenses to become short and compact and gains a typical chromosome structure. In metaphase II, the chromosomes align at the equator and the spindle fibers attach to the respective sister chromatids. In anaphase II, the chromatids separate from each other and move to opposite poles due to the contraction of spindle fibers. Finally, in telophase II, the chromosomes decondense. The nuclear membrane envelopes the chromosomes and the nucleolus also begins to reappear. The chromatids form the daughter chromosomes at both the poles. Last but not the least, cytokinesis helps divide the cell into two. Since two cells have simultaneously undergone meiosis II, we get 4 haploid cells at the end. The haploid cells are also called gametes and are genetically different from each other. And why is that so? Genetic difference mainly occurs due to recombination between the chromosomes, which takes place during meiosis I. And with this, we come to an end with the concept of meiosis.
Have you ever wondered why children do not look exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase II, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells.
The simpler one is mitosis, which produces 2 identical cells with exactly the same genetic information. The other process is meiosis. It is a much more complicated process, creating not 2 but 4 cells with only half the number of chromosomes, and crucially, all genetically different from each other. Both mitosis and meiosis include the same phases: prophase, metaphase, anaphase, and telophase. Except in meiosis, they happen twice, so they are usually referred to as 1 and 2. As always, cellular division starts with a process called DNA replication. This involves making 2 identical copies of the original DNA molecule. The cell ends up temporarily with double the normal number of chromosomes. You can learn more about DNA replication in this video. In prophase I, the duplicated chromosomes join up with the pair from the other parent, so the mother's pair bind with the father's pair, forming a group of 2 chromosomes called homologous chromosomes. What happens next is vital for successful meiosis. As each chromosome is lined up next to its partner's pair, one chromatid from each side gets entangled with a corresponding chromatid from the other side. This is called crossing over. During this brief period, the 2 chromatids swap certain sections of DNA. This is called recombination. The sections that they trade correspond to the same location, so that each chromatid retains the correct number of genes. Recombination is really important because it creates variety. The new cells aren't identical to the other parents, and they also are different to one another as well. They are new genetic combinations. In fact, that's the whole point of sexual reproduction. It increases genetic variability. Each chromatid is now different, and as each one will end up in a separate gamete, it means each sex cell is genetically different from all others. Next comes metaphase I. As the chromosomes align themselves up in the middle of the cell, and in anaphase I, the spindle fibers pull the chromosomes apart to opposite ends. Then during telophase I and cytokinesis, the cell pinches apart in the middle, and the nuclear membrane reforms around the 2 new daughter cells. That's the end of meiosis I. Now for part 2. We start with our recombined daughter cells, each still with 46 chromosomes, but sperm and egg cells only have 23 chromosomes, so we need to cut these cells in half. Here goes a second round of division. The process is exactly the same as before, except that there is no DNA replication. We start straight with prophase II, with chromatin clumping again to form chromosomes. Then a line in the middle of the cell during metaphase II, and chromatids are pulled apart during anaphase II by the spindle fibers. Telophase and cytokinesis pinch the cells together, with 4 new granddaughter cells being formed. The end of meiosis gives us 4 different sex cells, each with only 23 chromosomes, ready for future fertilization. But that's a story for another video.
Meiosis involves a single round of DNA replication followed by 2 cell divisions, meiosis I and meiosis II. The entire process of meiosis results in the formation of 4 haploid daughter cells with half the chromosome number as that present in the diploid parent cell. However, after fertilization occurs, the chromosome number gets restored. Meiosis I includes 4 stages: prophase I, metaphase I, anaphase I, and telophase I. Let's know about the homologs before studying the phases of prophase I. Each diploid cell has 2 copies of chromosome, one inherited from the male parent and the other from the female parent. These 2 copies, coming from different parents, are similar to each other and are called homologs. Now let us look at the different stages of prophase I in detail. To begin with, let us learn about leptotene. During leptotene, the chromosomes condense and become visible. The zygotene phase is followed by pachytene. During pachytene, the chromosomal crossing over now takes place between the non-sister chromatids of the homologous chromosomes. This crossing over is what makes each individual unique and causes the recombination modules to be visible. As the crossing over completes, it marks the end of the pachytene phase. Diplotene is followed by diakinesis, which is also the last stage of prophase I. During diakinesis, the chromosomes are fully condensed, the nucleolus and the nuclear membrane dissolve, and the mitotic spindle begins to assemble. Prophase I is followed by metaphase I. During metaphase I, the microtubules from the opposite poles attach to the chromosomes at the kinetochores, and the bivalents align along the equatorial plate with a random orientation. Either of the parental homolog takes up any site on the spindle. The metaphase I is followed by anaphase I. During anaphase I, the homologous chromosomes separate out and move to opposite poles. With shortening of the microtubules, the sister chromatids, on the contrary, remain attached at the centromere